Hello, can everybody hear me? Okay, so well, good morning and good afternoon, everyone. It's about time to get our webinar started. So first of all, welcome to Novogene Genomic Research Live webinar today. My name is Wei Lin, Novogene's account manager based in the Netherlands, and I'm today's host. Today's webinar will take about 45 minutes, followed by about 10 minutes Q&A sessions, and we'll round up by a summary. So first of all, let me switch on share my desktop. Well, Novogene is a global leader in next-generation sequencing, founded in 2011. So we have remained at the forefront of innovation, delivering world-class services to advance discoveries in life science, healthcare, and even beyond. With the state-of-art laboratories and operational site across the United States, the United Kingdom, Germany, China, Singapore, and Japan, Novogene has established a strong global presence to support scientific and clinical advancement worldwide with highly skilled and interdisciplinary team. Novogene services covers the full spectrum of multi-omics from genomics, transcriptomics, proteomics, and even extended to metabolomic services, empowering research and organization with cutting-edge multi-omic solutions, supported by our high-throughput sequencing platforms, automation workflows, and comprehensive bioinformatic analysis. Through extensive collaborations with leading academic institutes, hospitals, pharmaceutical companies, and agriculture enterprises, Novogene continues to shape the future of multi-omic research and precision medicine as your trusted partner. Well, today is our great honor and privilege to have Dr. Jingyuan Fu here to get inspired by her prestigious microbial functionality study, which was recently published in Nature Review Bioengineering. Dr. Fu is the professor of system medicines at Genetic Department, University Medical Center, and she's also the member of Royal Netherlands Academy of Arts and Sciences. Welcome, Professor Fu. Professor Fu has led a groundbreaking research that systematically integrates large-scale multi-directional omic data on human genes, gut microbiome, environmental, and lifestyle factors to unmask individual heterogeneities in health and diseases. Her research focuses on interaction between human and microbe in regulating human metabolism, immunity, drug effectiveness, and safety. Moreover, she has been collaboratively developing advanced liver-on-a-chip models, which associate bacteria cultures with organ-on-a-chip technology to simulate microbe host dynamic in vitro. Furthermore, as the co-founder of the Groningen Microbe Hub within the UMCG, she also accelerates the transition of scientific knowledge to practice. The human metagenome comprises the collaborative genomes of microorganisms. Since the middle of 2010, the application of culture-independent high-throughput genetic sequencing technologies has promoted the expansion of the human microbiome database together with substantial advances in bioinformatic tools. However, the functions microorganisms can perform remains largely obscured, and the potential of metagenomic data to reveal microbial functionality has not been fully unlocked. To unravel those riddles, Professor Fu will be presenting today an overview of metagenomic data analysis from 1D species identification to 4D studies of spatial temporal dynamics. Professor Fu, please, the stage and the microphone are yours.
Yeah. Thanks for Dr. Liu's introduction. I'm going to share my screen right now, so hopefully you can see it in a second. So, yes. And again, thanks to the very nice introduction. And it's also my greatest pleasure to give this webinar at the Novogene and Symposium. And sorry, what? This slide doesn't move. Okay, strange. Okay, yes, sorry, the slides doesn't really move from my side. And over the past few decades, as we know, next generation sequencing and technology has completely transformed the human genome research in many different ways. This is especially true for the studies of the human gut microbiota, the vast community of the microorganisms that live in our intestines. Most of these microbes are anaerobic. They are very difficult to survive out of the human body and very hard to grow in the lab. But thanks to the modern DNA sequencing technology, we can now just direct extract the DNA and sequence them from the stool sample and find them what microbes live in our gut and what function they perform. So this technology breakthrough has made microbiome research not only a fascinating area for microbiologists, but also an exciting playground for researchers like me, who work and has background in genomics and bioinformatics. My primary research interest is to understand why people differ so much in their disease and sustainability and therapeutic responses. To answer that question, I use large-scale prospective population-based cohort called Lifelines from the northern Netherlands. This cohort, including more than 167,000 participants and covering almost tens of the population from the northern Netherlands. Every 1.5 years, participants receive all kinds of questionnaire regarding their disease diagnosis, lifestyle, medication usage, and living environment, job, economic status, and so on. Every 5 years, they went to Lifelines Clinic Center. So we collected different kinds of biological samples, including plasma, urine, fecal hair, and also Lifelines clinicians also measure all kinds of the functions. So through this way, we collected a huge amount of biological samples, as well as a large collection of the data, and including over 2,000 different phenotypic traits from each participant. So you can imagine it is a very large data and a biobank located in the Netherlands. And of course, this is only just on the epidemiological study and having this cohort individuals samples collected. But based on this cohort, when we have carried out several add-on studies to enrich genetic, microbiota, and various omics datasets in several subsets of the participants. One sub-cohort, we call it Lifelines Deep, and which contains 1,500 individuals, where we have a very deep omics datasets profiled, including genetic, microbiome, transcriptome, proteomics, metabolomics, and so on. So the gut microbiome data was collected first at first time point was in the year of 2012. And then followed a second time point was followed up on another 5 years, 4 years in 2017. The connection, the third time point is also finished, is under an investigation, and the fourth time point also started and almost finished as well. And based on success of the Lifelines Deep cohort, at the second time point, we also expand our microbiome research to the Dutch Microbiome Project, where we connected the fecal and also nose and tongue and oral swabs for 10,000 individuals. And the microbiome data has been analyzed. Now there also some studies are still ongoing to study also the microbiota in the nose and in the oral cavity as well. So now, and also followed upon for the second connections, and the third point is also ongoing. As we also know, and because gut microbiome is established and in early life, and the early life establishment is so important for the later health. Based on that, we also initiated the lifelong next cohort. We are following 1,500 pregnant women and newborns for the multiple and with many time points during pregnancy and also several time points during the first year of life. Afterwards, this cohort will be also continued for the long-term follow-up. This project mainly initiated by my colleague of Professor Alexandra Janakova. But altogether, this is a massive project focused on gut microbiota and host microbe interactions. Human health had involved efforts for many PIs during the past decades, as the picture is showing over here. But it's also nice to mention that metagenomic sequencing for our Dutch microbiome project was carried out by the Novogene. The study was published in Nature in 2022. In the abstract, we mentioned we had analyzed, microbiota composition, function, antibiotic-resistant valence factors in 8,000 Dutch resident individuals in a three-generation cohort composed of 2,700 families. In the method session, you can also read metagenomic sequencing was performed at the Novogene China using Illumina HiSeq 2000 platform that generated 8 GB data and for each samples. So this is a very successful and collaboration between us and the Novogene, and we have been very pleased with the sequence quality and also the effective collaboration, especially for such a large-scale project. However, perform metagenomic sequencing is only the first step in the microbiome research. The real challenge lies in how we can extract meaningful information from the sequencing data. Recently, we proposed a multidimensional metagenomic framework that integrates state-of-the-art bioinformatics and experimental techniques from 1D to 4D analysis. So in the coming 30 minutes, I'd really like to present our recent studies also lined along this multidimensional metagenomic analysis framework, and also show how it works. The first 1D is we call it refer to species identification abundance profiling. It is because you can characterize these things even in one single sample. So there's many approaches has been identified. When you're from the metagenomic sequencing data, you got raw reads. So you do the quality check and filter out your raw reads and then get the cleaned reads. Using cleaned reads, there's 2 ways you can move from your high quality reads to the taxonomic information or even functional pathways, particularly based on metagenomic sequence data. One approach is a reference-based approach that many tools have been developed. If, for instance, by Bakery and toolset, you can use them for each species. They have identified 100 to 200 different marker genes. You can align your reads to those marker genes, therefore quantify the abundance of each individual species. Another way is what we call called reference-free approach. In this case, the raw reads, clean reads will be assembled together as a contig and further constructed as metagenomic assembly genomes. Based on these assembled genomes, you can align your reads to assess the abundance. So this allowing you to have your own cohort-specific, sample-specific genomes to be analyzed. So it's called a reference-free approach. So many tools has been developed from both reference approach and non-reference approaches. Both have certain pros and cons depending on your research purpose. So normally reference-based approach is faster and also easily compare across different studies, and a reference-free approach allowing you to identify very rare and specific taxa properly specific for your samples. By applying 1D level, actually, most of the microbiome research are very familiar with. And by doing that, you can profile the taxonomic composition for each of your samples. In this case, for instance, I show in our microbiome composition and for our Dutch microbiome project, either in the final level or in the family level, but of course, with metagenomic sequencing data, you can either extract the abundance information for species and even in the upstream level. And you can see, and actually microbiome composition diverges greatly between different individuals. In this case, some people had a very high abundance of firmicutes, while in some individuals, their gut microbiota is dominated by Bacteroides. So this inter-individual variation can be influenced by many factors. Based on the family structures of our samples, we can see comparing unrelated individuals, the individuals living in the same household, like partners, the microbiota similarity is higher, that here in the distance is smaller. So the distance is even smaller between parents and the child, and even further smaller between between siblings, brother and sisters. So this indicating microbiota differences is driven not only by environment, but also by host genetics. We can further quantify, and for each different species, how much variation could be explained by genetics, here indicated by the blue bar, or by environmental factors, here by the green bars, or by core housing, where we can not perfectly define it due to genetic or shared household environmental factors. Generally, we can see an environment dominance over genetics in shaping our gut microbiota. Of course, this inter-individual variance also relevant for human health. So because our study is a population-based cohort, therefore they contains different diseases. This disease prevalence very well replicate the overall prevalence of the common diseases in the Dutch population. So we did a systematic analysis, and for different kinds of diseases, if we had sufficient sample size, such including cancer, cardiometabolic disorders, gastrointestinal diseases, neurological disease, and other immune-related diseases. You can see many different species are shared across different diseases areas. Interesting, even we found one species, if show high abundance in, for instance, in the cancer, the species also showing high increased abundance in patients with cardiometabolic disease or gastrointestinal disorders. So this suggests that even people have a different disorders, it seems the gut microbiome shifted to the all similar dysbiotic status. But actually, we're also able to find this shared microbiota dysbiosis cannot simply explained by disease comorbidity, as we show in the lower part indicating the sharedness of the microbiome across different disease. The upper triangle of this correlation matrix indicating the disease comorbidity. You can see the sharedness in the diseases microbiota dysbiosis significantly stronger than the disease comorbidity. So indicating, though we may have different diseases, but actually underlying microbiome disease etiology related gut microbiome very likely to be shared. So this is, however, we are not based on abundant species. We're still not able to tell the functionality. Because even within species, same species, there's a very big genetic variation, and also at strain level, we already know they have a very big functional differences even at strain level. So this raises to another level of analysis that we call it 2D dimensional analysis to assess strain level genetic variance, even even between within species level, same species level. This is because even within one species, they could have many different kinds of bacterial genomes. And by comparing the genome similarity of the across within the same species, some genome part are shared across different genomes with it's called core genome. However, there's a big part of the genomes only shared by a small proportion of the certain genome or even specific to one particular genome or particular strain. So we call it share or cloud genome. So marker genes basically used identified from the core genome that all the genomes from same species all shared. We are not able to answer the questions, refer the question on functionalities that are representative in the share or cloud genome. So this is raised importance actually look into strain level of genetic variation and functionality. Even with that one, we can even do the most individual genome analysis and genetic variance analysis. One genetic variance could call a single nucleotide variance. That means the polymorphism at the single nucleotide level. A lot of type of genetic variants could be related to a very large genomic region that can be either duplicated in the genome or completely deleted in the genome or a reverse. We call it the structural variation. So they have a variable structural variation, deletion, or inversion. So bring that in mind. We first collaborated with Professor Nicola Segata's group from Italy and performed a genome-wide, global-wide analysis to investigate microbiome genetic diversity across 33,000 metagenomic samples from 42 different countries. Most of the samples indeed from the European countries, but we do have a substantial amount of samples from North America, South Asia, Africa, and even from Asian and South American samples. So from these samples, we first constructed a phylogenetic tree for each of the samples. We call it a metagenomic assembly genomes for each of species. So for those species, we were able to construct it for a totally 160,000 species. But as you know, not all, all species has a sufficient sample size. So we first calculate the SGBs and prevalence across all the global samples, and also look at also the per tree, how many samples we have for individual trees. And so we can see over 500 XGBs, we could have over 300 samples that actually we follow up on this analysis, mainly for those trees, these SGB species. As you see, but that refers to red dot in these pictures, the taxonomic distribution of all the SGBs. And you can see on those 500 SGBs actually very well presented for most of the taxonomies that we're able to identify in the human microbiota. So this is one example. This tree is phenotypic trees, one particular species. So in this case, one branch indicating one sample. And then the distance between the different samples reflected how similar in their genetic makeup. And then we can align the different phenotype of these individual samples to this tree. We use the phylogenetic generalized linear model, mixed model, to map in the tree distance with the phenotypes. The first thing, the logical thing, because we had samples from different continents, is also very well known that there's a geographic effect in gut microbiome due to either host genetic environment, exposure, and lifestyles. So it's very logical and not surprising to see actually different samples from different continents indeed co-mapped with phenological distance of these trees. You can see the samples, for instance, from African samples Asian samples or North America or European samples are clustered together, indicating and also aesthetic, also lifestyle coherence. Actually, this phenomenon is not rare. It's not only observed for this particular species. Actually, we're able to find out of 583 HGBs, we found a significant geographic effect for 456 of them. And this also very well support the previous observation and published in Nature a couple of years ago, says the core diversification of the gut microbiome with humans. So with during the long-term, evolution history, not the human genome of lifestyle, environment exposure, also gut microbiome may follow in similar evolution and development and stress. However, even after correct geographic effect, we're still able to find some particularly phylogenetic clades associated with host phenotypes. For instance, I showed here two examples. Here is a particular clade, and in both in Asian samples or European samples are enriched for the elderly individuals. And this is Japanese samples in the Caucasian individual, and here is a more elderly individual from European samples. And actually, they come from the same phylogenetic tree, just try to demonstrate the same branch. We divided them and extracted them from the both Asian and European countries samples. So you can say actually they are not overlap with the geography or region of the samples, particularly clade was enriched in the elderly individual, and also we found that this particular clade also had a distinct bioassays, relevant bioassays metabolism. Another clade is over here in the Cornicella allefaciens clade. It was found enriched for the patients with melanoma and prostate cancers. Both European and also American and also Asian countries samples are all clustered together and also found indicating maybe there's some specific functionality related to cancer patients. Though we cannot tell even for this analysis, we're only able to show association. We don't know it's really the causal and the causal inference analysis and also the consequence relationship. This also indicating we have to maybe zoom into genomic region for those particular clade and whether we can help us identify functionality. That's also the reason we not only do the phylogenetic tree-based analysis, we also do association on the individual genetic variance analysis level. For instance, case here, I showed you on structural variation level. Structural variation, as I mentioned, refers to the larger scale region, genomic region that either be duplicated and or deleted in the microbiome genomes. Here we focus on two types of structural variation, which is variable structural variation or deletion structural variation. And we found this structural variation is very common in the bacterial genome. In the previous analysis, we already have identified over 8,000, nearly 10,000 structural variations in about 100 different bacteria species. By filtering out and focus on the common structural variations here we focus on 3,000 structural variations that presented in over around 60 different common species in the gut microbiota. And one study, and we did actually recently study published last year, and we linked the structural variation of the gut microbiome to host genetics. As I said, if we see the core the evolution of the human and gut microbes, are we able to find a genetic link between the host genome and the gut microbiome? So actually, we did find quite a lot of signals, but the one signal is very striking. That is located APO locus, refer to the genes actually decide the blood type of host. And this APO locus determines the terminal oligosaccharide blood type antigens. If you have an A blood type, and then that means your terminal organoid is called a GAG-like. So if terminal organoid is a galactose, then you have a B blood type. If you have both of these oligosaccharides, you have AB, but if you have none of them, you have O blood type. As we know, these blood type antigens actually can be cell-free. Dependent on the genotype of FUT2, they can be secreted to the intestine. So then in the secretors, FUT2 secretors, these antigens will reach to the epithelial mucus. If you're non-secretor, and then you will not. So then we think, okay, then we believe in, because of that, then we hypothesize the APO association should also dependent on the FUT2 secretor. So we further did analysis, stratified analysis based on FUT2 secretor status. Indeed, we found the association was particularly enriched with A blood type and in the FUT2 secretors. If it's non-secretors, then this association is not significant. Then, because we now know which genomic region is relevant to a blood type, and then we can zoom into that region to identify which genes or pathway relevant underlying this association. So we isolated 12 different strains from fecal samples, and 7 of them with this strain and with this region, and also 5 of the strains without this region. By these isolates of those individual species and strains, we also found actually our bioinformatics analysis only captured here this region, smaller region of this structural variation region. By looking at the isolated whole genome, and then we are able to extend the structural variation to a much larger region that contains 21 kb. Then we did the gene annotation for the whole region and identified a completed pathway that involved the GAG-like metabolism related to catalyze the A-type related terminal oligosaccharides. So I really want to highlight actually this association, APL association for the Faecalibacterial parasites, which we identified, not able to see in the species abundance level, complete up, exactly same data set, same cohort, and so exactly same sample. At the species abundance, we not see any association. Then at structural variation level, we do find a significant association, 10 powers minus 42. But through structural variation, we identified the candidate gene. At the gene level, we can find that the p-value has been significant. P-value has reached to 10 powers minus 163. So this is also really indicating, even with the same genomic sequencing data, depending on which level of information you can retrieve, you can find different information. And also don't only look at a species abundance, also look at the strain level and the genetic variants would able to help us to extract the true informative information. Of course, we do not also end up with that. We also perform the function analysis to prove and validate our observation. So we found indeed the strains with that GAG-like pathway indeed can grow with the media with GAG-like as the solar energy carbon source. With species without this pathway cannot grow. But if you had sharp eyes, you may notice, oh, there's also one strain marked as red, and means that it has the gut-like pores but cannot grow in that gut-like media. What happened to that strain? Actually, we found that this strain is though they had able to map that region, but then we found that this region was completely flipped. So it's a lot of structural variation I didn't talk about is reversion. That is indicating this genomic reversion has deactivated the gut-like pathway. Therefore, this strain cannot grow with the gut-like media, reached media. And it suggested that indeed, this diverse can influence the functionality. Another observation we found also we pity that we have missed it. And we identified that the first step of this pathway actually is carried out by the GH109 gene, which help to cleave the terminal oligosaccharide from the broad type antigens and free the oligosaccharide to the uptake by the transporters for downstream metabolism. So you can imagine, as I mentioned, the terminal oligosaccharide can determine the broad type. So if you cleave the gut-like from the broad type A antigen, then broad type A antigen could become to O, broad type O. So this was so cool because a few months later, another group published a paper in Nature Microbiology to indicating how we can utilize those bacteria, these enzymes to produce universal broad type 2 transfusion. So really indicating a very significant clinical implication. So it was so cool. So, and now we talk about 2D level. But keep in mind, in base metagenomic sequencing data, we can basically see, okay, which enzyme we have, and in this, which genes have carried out bacterial enzyme. But most of those genes actually still poorly allocated. We still don't, I think more than 80% of the genes still lack of allocation. So then the next step, we also propose how, also not we, also many other people as well, how we actually to use also 3D, leverage the 3D structures of the proteins to enhance our function prediction of unknown proteins. And many of you may have aware of the big AI breakthrough in structural biology. For instance, AlphaFold and has been released a couple of years, had a great large language model, has greatly accelerated the structural biology and development. And a couple of years ago, they also had a big atlas has been constructed called evolutionary scale modeling atlas that can already generated 3D structure prediction for 772 million proteins derived from metagenomic samples from the variety of organisms and environments. They also constructed a 3D structure network, a similarity network, to facilitate the function prediction and also interaction with other molecules. So this also inspires us whether we can use 3D structure to annotate, identify novel microbiome genes. And its answer is indeed true. So previously, we also did the structural variation analysis with bioassays. We know bacteria involved in secondary bioassay metabolism. Through this analysis, we do able to find certain structural variation regions harbor no genes. For instance, in this case, by previous identified bioisohydronase genes, which are associated with the primary to secondary bioassay ratio, because these genes indeed involve the first step of the transformation from the primary to secondary bioassays. However, we also identified some structural variation region which contain not previous unidentified region. We don't able to find in this region any genes previous known to be involved in the bioassays metabolism. But by leverage 3D structure and sequence analysis, we're able to find actually one gene here actually with unknown function, unknown name, which very likely to be to the bi-ISD hydrogenase. Because we know these genes, the green ones is from the known genes from the E. coli. The orange ones are from the unknown genes from this particularly species, Rhinococcus species. And it's also very similar structure, 3D structures, and also exactly at lattice sites. So then we can say based on 3D structure, we're able to annotate these unknown genes to be likely to be an HSDH gene. But on the other hand, we're also able to, even we're able to know the gene, but we still don't really know which metabolites these genes can be involved. So then a lot of efforts we made actually to based on known gene, whether we can predict unknown metabolites. This is a method we developed and published early this year. We call it MicroRS. It is an enzymatic reaction-based metabolic prediction tool. In this tool, we composed 12,000 metabolic reactions from the human genomes and over 8,000 unique reactions from 6,000 different bacteria strains. And this how MicroRx works, and it has several modules. Firstly, we constructed reaction rules. This to simplify the metabolic reactions and to the different level of to the reaction rules with different level of confidence levels, depending on how many extra items next to the active chemical groups. This is the orange part indicating the crucial reactive groups, chemical groups involved in the chemical transformation. And the more items you add in the action rules, it become more specific and to the reaction, but also less flexible to the novel prediction. Secondly, we also had added several analysis modules to analyze on the metabolites itself to see how chemically, structurally, how reliable it is, also on the genomic levels, and to say how those involved enzymes, genes, how comparable across different species, and also 3D structures, how convincing or not by the binding accessibility and molecular accessibility sites. And as we previously know, gut microbiome has also involved in the influence drug efficacy and toxicity. One example, for instance, an L-DOPA, levodopa for the treatment of Parkinson's disorder, gut microbiome affected it is about availability because bacteria can convert L-DOPA to the dopamine, actually this active mode of the drug already in intestine. Actually, we wish to get a dopamine transmission happen in the brain, but not in intestine. If it happened in the intestine, of course, bioavailability is reduced. And even worse, dopamine can be further converted by bacteria to other compounds which actually exist toxicity. So based on this one, people also defined the compound to try to inhibit this conformation, target the gut microbiome, thereby improving drug bioavailability by inhibiting gut microbiome. Another example, of course, gut microbiome can influence drug toxicity related to the anti-drug cancer treatment. And bacteria can convert the insilent metabolites SN38G and come back to the active mode SN30G in the intestine, therefore existing toxicity towards the epithelial cells cause severe diarrhea. So then we think how using our MicroRS to predict which drugs can be metabolized to the gut microbiome, which cannot, or which show which type of metabolites can be can be carried out. So therefore we carried out a systematic analysis using applying microRS to over 1,000 FDA or approved orally administered drugs and for the prediction. And we can find this is the drugs we have been predicted, and you can find some drugs can only metabolized by the human enzyme. Some drugs found only by prediction, only metabolized by the gut microbiome, but we do find a big proportion of the medication can be metabolized by both human enzyme as well as gut microbiome, highlights the importance of the co-metabolism of host and gut microbiome in drug response, and indicating in the next level of personalized medicine, we also should take gut microbiome into consideration. To give you one example, for instance, the tryptophan metabolism, and we found many drugs have we involved conversion these subchemical structures, and we know that many drugs, for instance, the I think it's a tryptophan, is for the drugs used for treat for the migraine, and also melatonin treated for the sleep disorder, and also anti-drug cancer drugs all had these similar chemical structures, suggesting these enzymes can be also involved in transformation of all these different drugs. Interesting, we found that these enzymes not only presented in the human genome, but also found it in 107 different bacteria species. By looking at the structures of these enzymes across different species, you can see they show very high similarity in the strict structures as well as in the catalytic sites. So this is a 3D level, but as we know, gut microbiome different from the human genome, and is more spatial diverse and also temporally dynamic. As all human cell types may have the same genetic information, when we're talking about germline variants, of course, and which are generally remain stable over time and not changed at all unless some somatic mutation occurred. However, gut microbiome community and show very big differences in the different part of your GI tract, and also we know, and also terminally big variation over time, as we see a lot of variation in the infant, in the elderlyhood, and also in elderly and in the aging individual as well. So this also indicating simply by sampling, cross-sectional sampling microbiome samples from fecal sample is insufficient to understand host micro interactions in human health, in health development and so on. So this requires large-scale human cohort study with longitudinal sampling and even novel techniques, for instance, capsulate, allowing us to capture microbiome samples inside our GI tract. Also further technology and, for in vitro models like organ on a chip to assess and host microbe interactions. So the next level 4D analysis to understand the spatial temporal dynamics of the host microbe interactions in human health really require not only metagenomic sequencing, but also more advanced other high throughput biotechnologies together. Previously, in the mouse study, of course, we already had evidence to showing, by staining and in the different part of the intestine part, by staining, you can see the human cells, mucus levels, and also in microbiome distribution content are very different. More recently, by actually slice the mouse intestines and developed a novel technology called the spatial host sequencing technology. They can actually, one sequencing, they can capture both host RNA-seq for the host cells, as well as 16S of the microbiota to look at enable simultaneous capture of the host transcription and also bacterial community along the GI tract. But of course, these techniques can only be done for the experimental animal models, right? Because you need to get intestine and slice them in the different slices to the analysis. It's very difficult to do in the human cohort. But nevertheless, based on the fecal longitudinal sampling of the fecal samples, and we're still able to assess the temporal dynamics of the gut microbiota in human health, and also move us more closer from associated causality. This is indeed what we indeed did based on the lifelines cohort. For instance, by following 338 samples over 2 time points, we're able to assess the inter-individual variations within the same individual variations over time and compare it to the variation across different individuals here. We found we do find some species so large variation over time, but in general, there's a variation between 2 time points still smaller than the differences between different individuals, showing particularly for some species here down in lower part, they show very high similarity, basically across stability over time, very little variation over time, but very large differences between individuals. So this part, we call it maybe used as a fingerprint for the individual, indicating microbiome individual specificity. So it was indeed the case based on this, make use of these features, we can use a microbiota to identify the longitudinal samples whether belong to the same host or not, with accuracy and with 99% of specificity and 88% sensitivity. Our longitudinal sample spanned 4 years apart. We applied this model for the human microbiota project that the fecal samples was collected at 2 time points with 1 year difference. You can see this specificity and sensitivity much higher. That, of course, is logical. The shorter duration, the more stable your gut microbiota is. But on the other hand, on the other pictures, we do see some species exist a big variation and temporal variation. And these features may allow us to move from association to causality. And for instance, for the fecal bacterial parasites, as we all know, is a beneficial one and a famous butyrate producer in our gut. By looking at the genetic makeup of fecal bacterial parasites, we can identify 2 big clades. Let's just simply call it 2 strains. Strain 2 is more common and presented over 75% of the samples, and strain 1 less common and 25% of the individuals. Look, by following the timeline, and then most people keep the same strain over time. 4 years later, most people still have the same strain. However, over 30 individuals, strain replacement occurred. 24 individuals had the strain 1 at the baseline, but had strain 2 in 4 years later, and vice versa for 13 in other individuals. Interesting, we found that different strains associated with different metabolites, particularly percrasal sulfate. The strain 1 seems not a good one because percrasal sulfate is derived from bacterial amino acids, had a toxicity effect, eliminated through the urine, so it's a kind of the uromic toxin. And the higher level with the individuals with strain 1 and showing a higher level of percrasal sulfate in their blood. This based on this association. But if we look at the transmission individuals, the individuals who transmit from strain 1 to strain 2, for those individuals, we found actually the percrasal sulfate levels was decreased over time. And vice reverse, if individuals move from good strain 2 to the bad strain 1 here, and the percrysal sulfate levels also increased over time. So through this approach, we not only establish association, but also provide evidence for causal link between these metabolites and also different strains of the fecal bacterial parasites. Of course, we do this analysis not only for the metabolites, but also able to do this analysis with host phenotypes to construct a mediation analysis to see how the changes in the microbiota species or the metabolic pathways influence the changes in metabolites, thereby contribute to the changes, in this case, mostly related to cardiometabolic phenotypes, including blood pressure and glucose, lipids levels, glucose levels, and so on. Altogether, I think I gave a little bit of overview and combined with our the data from our study to showing how by utilize the metagenomic sequencing data with also various level of omics or phenotypic data, how we're able to extract information from 1D, composition, species, abundance, to 2D, genetic variance, phylogenetic distance, even to the functionality, 3D, to identify novel gene and also novel metabolites, as well 4D, to establish causality and understanding temporal and the dynamics of the gut microbiome and relevant to human health. At the end, I also like to acknowledge, thank my group members and also funding institute that support my research. Thank you. I would like to take some questions from now.
Well, thank you so much, Professor Fu, for your very comprehensive and insightful presentation, covering from identification to variation diversity and to protein metabolite structure, eventually to another dimension, spatial and temporal. So we are entering Q&A sessions. The first question is that how statistically challenged you have faced during your, let's say, population-based study, and how do you tackle those challenges?
Wow, this is a big question. I think we can give another 2 hours lectures regarding different challenges. And the first simple way, we start with previous established methods. A lot of beginning and approaches we used normally following the technical bioinformatics pipelines has been previously constructed, for instance, by query tool set and so on. This is starting point. And then secondly, you need to keep your mind open, and not all the messages, not stop that I showed. For instance, first one is only look at species abundance. Then you also need to aware what bring your information, biological questions in your mind to think whether the information you retrieved based on first step of the analysis able to answer your question. You need to have a very good understanding of your data itself. And the first challenge, indeed, then you are starting to think whether your pre-cleaning process is right or not. Secondly, whether you are right and retrieve the right information at the right level as well. That I said, if I do the analysis only focus on species abundance level, or I do analysis on the genetic variant level, even the same data sets can actually yield very different results, even not significant or not significant results. You may even miss information there. And so this is on the biological level, the challenge is just think how you would like to retrieve information. Secondly, is a technical challenge. And technical challenge is very big. It's related to very computation challenge, whether you had that capacity to handle this large scale data, particularly in our data. We had over 10,000 samples, even globally, we had different countries, and whether you're able to handle it. And also the data, also, as I said, it could come from the different studies. If you have your own study, probably the technical variations is less because we harmonized in the experiment level already. But if the data from a different cohort, different DNA assimilation methods, different sequencing methods, how you're able to combine them together. and how you need to do a lot of a benchmark. So this is not a this the challenges really have to define in the different, so many different levels you need to think about. And the final level is association level, and whether we can, what is the right analysis model to perform association. And what is covariance we need to take about, particularly gut microbiome, is so complex. It can be influenced many different phenotypes. Not every cohort, for instance, has a diet, has environmental factors, as such extensive phenotype that we have collected in lifelines, but maybe not missing. Now, how we manage the richness or difference in the information in analysis model. So I think the challenges is also a solution is rather dependent your cohort, the level of the studies, and also the biological question, level of the biological question you want to address.
Yeah, well, thank you so much for your expertise. Of course, we can further extend discussion, but yeah, perhaps we can, let's say, our audience can email Professor Fu for more detailed discussion. The second question is that how do you envision the microbial fingerprinting becoming a part of personalized health monitoring or for clinical diagnostic? That's another big question, I would assume.
Yes, I think this is a very good question and also very relevant. I think this is also our dream to implement gut microbiome in personalized medicine. And human genetic has been already very well implemented, particularly, for instance, I don't know whether any of you have heard the pharmacogenetics passport project. You can get your your genetic information based on genetic variants of the important for drug metabolism. You can hand this your genetic passport to the pharmacist and tell you should use this drug and other drug. This is a very easy and straightforward approach, and because our genetics generally not change. But the gut microbiome, as I said, as I showed you, it seems has been its role in personalized medicine has seriously overlooked. This however, is also reflected how difficult it is, though it's very high, despite of its high potential. So we need, because this dynamic change, right? We need to constantly, not like a genetic. If I had the sequenced genetic passport done once for your lifetime, done. But you need to constantly monitor the gut microbiota to build a very specific microbiome fingerprint for health. So that is a very challenge. And you need a constantly monitoring. I was hoping, because only some people already talking about smart toilet. People may think it's a fantasy, but I do believe such a smart toilet should be maybe become reality in the near future, because I know it's happened, and in the clinical setting, in a company in the US already made such a smart toilet for the clinical setup. Even in our UMCG, seems in the plant also have those smart toilet in the gastroenterology department. Patients should come and develop food. We can immediately get the sample stored and sequenced, running simultaneously. It will be super cool. Maybe in the future for the household. Secondly, as I showed, microbiome dysbiosis seems show higher similarity across different diseases area. So I feel we still lack sufficient knowledge to understand specificity in related species. Based on gut microbiome, as far based on evidence I have seen so far, we are able to tell how likely you're healthy, unhealthy based on microbiota, but still with this information is not specific enough for me to tell you which type of disease you're going to have. This also happened in many studies compare diabetes, for instance, to healthy control. heart disease to health control. Everybody compare the patients with health control instead of compare across different patients. So I think the next level of information or analysis vision, we should also have develop more disease-specific biomarker in the gut microbiome. Yeah.
Okay. Yeah, I really appreciate for your sharing your vision. And then the third question is from Ashika. So she asked, can 2D be done only for large global variants, or it can be done for same species? For instance, 6 different individuals from a study organism.
It's you can anyhow, even two samples, you can do the differences, But
Mhm.
the point is that how this difference is meaningful for you. That is the
True.
question. And do the genetic variance analysis require high quality of reads? So that's the reason we're able to construct, for instance, a phylogenetic tree or structural variation and for thousands of species. But we lose a lot of sample. For instance, for the phenetic global analysis with the 33,000, for some trees, the majority of the trees, more than 1,000 of the trees, we have the samples with less than 300 samples out of 32,000. The reason for that, we need sufficient reads covering the whole genome for that particular species to enable to do that. And so that the big loss in the sample size per species level. So if with a small set of the study with a small sample set, you can do it. But unless you have a very deep sequencing, you must enable you have enough reads to covering all the species of interest to enable to construct the tree to able to call genetic variants. If you don't have a very good coverage on the whole genome level, this approach would not work.
Yeah, that's true indeed. It's about the statistic and the coverage.
Yeah.
So, well, unfortunately, due to time constraints, and we have to end up the Q&A session. And before we end our seminar, I would like to share several take-home message with you guys. To round up today's webinar, I would like to share several take-home messages highlighted by Professor Fu's study. So the first one is multidirectional microbiome profiling spans 1D composition, 2D genetic diversity, 3D structure, and 4D spatial temporal dynamics, offering a comprehensive view of microbial ecosystems. Artificial intelligence-driven metagenomic accelerated research by enabling rapid precise analysis of the microbial composition, function, and interaction, revealing hidden patterns in the human gut, which has not been unmasked before. And emerging technologies like spatial resolved metagenomics, single-cell sequencing, and organ-on-chips models, and together with the smart toilet that Professor Fu also mentioned, enable precise microbial manipulation, driving personalized intervention and novel therapies. The last thing I want to share is that AI-powered multi-omic integration synthesize multi-layer data to enhance biomarker discovery, clarify gut microbiome host interactions, and advance innovative diagnostics. So, sorry. Yes. So thank you so much for your time and presence today. And we hope that you enjoyed the webinar and found it very insightful. As mentioned, we haven't managed to answer all your questions, but we'll come back to you later by emails. And we will also email you a link to the webinar recording, since we saw the question in the chat box. And together with the slide deck, you can expect to receive the week after. So when the webinar finishes, a survey will pop up, and we appreciate you taking your time to fill it out, so as it helps ensure us make future webinars even more relevant to you and your research goals. To appreciate your attendance at the webinar, Novogene is pleased to offer you a 15% discount on your coming project. An email will be sent to you within the next few days, and the code that you can use to apply the discounts. Moreover, there will be a link in the follow-up email for you to review a certificate of attendance if you need one. So this can be printed out from your internet browser. So lastly, in the meanwhile, you can follow us on Twitters and LinkedIn to keep up to date with our future webinars, offers, and new services and news, of course. We're looking forward to seeing you all again on the next webinar. Have a pleasant day, and once again, our gratitude to Professor Fu for her comprehensive study and a wonderful presentation.